As we approach the end of unit number 1, I'd just like to say a quick word about material from the lecture and the lab. Inevitably, there's going to be a little bit of overlap of material. You'll see concepts and ideas, data from the lab that are also found in the lecture. When we have exams over the lecture material, I will work specifically from the material I give you during the lecture. Now, that also includes quizzes and reading questions, and then all of the stuff that we do for the videos for the lab lecture. Your SLOs are a great guideline. They will give you the vast majority of the material. If you master those SLOs, you should be very well prepared for the exams. Now, when it comes time to the lab, a lot of it is just doing the material, doing the reading questions, going through it, and we'll get more kind of technical as far as looking at data and assembling transcriptomes in the coming labs. And there will be a lab practical final. Again, that will be just because it's weird this semester, rather than having a practical one where you have to focus on the actual in-lab procedures, because this is all online. Your final lab will be taken from the lab questions. Some of those are ones that I'm having you answer and put online on lab questions. Some of them are the ones that you're just filling out in the lab itself. If you feel uncomfortable with any of those, because I'm not going through and grading those and giving you feedback on those, but if you feel uncomfortable with any of those lab questions, please make some time, send me an email, attend an office Zoom hour so we can review those, because it'll make it a lot easier for you if you're mastering that. You feel comfortable with those questions as you go along, rather than trying to play catch up at the end of the semester right before we get to that lab final. Okay, so we've talked about how scientists have been working towards this theory of an evolution of animal form. And this is kind of an all-encompassing theory, right? And a theory is a large body of scientific data that's been tested and supported, and we're very, very confident and sure that it's correct. And as we work more and more towards making our initial ideas about evolution of animal form at the genetic level into a theory, we're hoping to have an idea that will kind of give us a broad perspective over all ranges. So whether we're asking more focused questions like, why is a fruit bat different than this is more standard, this is North American, I actually think Central American bat. Why are these closely related species? Why are they a little bit different from one another? Well, we could go into that theory and begin to look at genes and patterns to answer that. And we could even then extend that to more distantly related questions. Why are these two arthropods? An insect here represented by Grilloblatta. A couple of you were assigned to look at this order of insect for the lab. Why are they different than a centipede, right? And we can look at these big picture questions in addition to the other ones. And as we have done this, as was shown in the reading that you did for the lab, there were some big surprises. Some major things that at the outset, right, our initial hypothesis just wasn't right on. So let's talk about some of those surprises. We'll look at the initial hypothesis, and then we'll talk about how Drosophila in particular, but study of other animals, has helped us to revise that initial hypothesis now towards a working theory, if you will, of animal development. Okay, so the first big surprise was that proteins often change dramatically over time. So as we begin to sequence genomes, sequence transcriptomes, map those transcriptomes onto the genome so we can figure out where exactly the genes are and how those are translated into proteins, what we found is that there were some remarkable examples of proteins that, although they had changed, that the function did not change as much as we might have expected based on how much the protein changed. And this goes back to a couple of things we can talk about. So neutral theory, which is a review, you've hopefully gotten some background about this in an evolution class. If not, neutral theory is the idea that the vast majority of mutations and changes that occur at the genetic level are effectively neutral, meaning that although we can map them onto the genome, we can recognize differences between species A, species B, that when it comes down to how did that specific mutation impact the organism that carries it, The answer most frequently is it didn't impact its phenotype at all. It's there in the genome, but that's not translated into the phenotype. Now, again, hopefully you have some background. There are a number of explanations for neutral theory. In my class on evolution, I go over 5 of them. I don't expect you to memorize or learn all 5 of them, but we're going to talk about one of them that is particularly important to this one. So we can have mutations that change the amino acid, maybe even change the amino acid sequence, right? Now, what I mean by that is maybe we have a mutation that changes a leucine to an isoleucine. And because those 2 amino acids are functionally very similar, although there's a difference in the amino acid at that one single residue, it might have no phenotypic difference at all in the protein. And so there's not any natural selection working to make that new mutation more frequent, which would happen if it was a beneficial mutation, or remove it from the population if it was a detrimental mutation. It's just neutral. And so we get diversity initially, and maybe then random fluctuation eventually fixes one or the other. And if it does that differently in different lineages, then we can have the exact same functional protein now with a single different amino acid residue. And sometimes there are even larger changes that might add a big chunk of a protein. But if it's occurring in a region of the protein that is not functionally important or is less functionally important than another one, it might be neutral or effectively neutral, right? So we can get single amino acids swapping in and out, and if that amino acid is the same, there's no functional difference in the protein. And more rarely, but still can happen, we can get larger mutations, maybe changing several amino acids, changing the sequence of amino acids fairly significantly without having an impact on the protein. Now, that doesn't occur all the time, but occurs often enough, and many of those are kept, and then random fluctuation means that we can see great variation in some proteins with relatively little effect on the function of that protein. And that really wasn't something that we thought about or understood because we didn't know much about the genetic code, about the degeneracy of the genetic code, which basically means that we can have redundancy at the DNA level, in changes at the DNA level that make no changes at the amino acid. And then also this range of different amino acids. We have 20 different possible amino acids that sometimes can have overlapping or similar function when they are put into effect in the protein. Okay. Now, not only that we can see as we analyze proteins that there can be changes in amino acids with little change in function, but there's this massive amount of gene similarity between organisms. And you've seen this before, and this can be misleading sometimes the way people present it. But you may have heard things like, oh, chimpanzees and humans have 90 some odd % genetic identity. Now, often you have to be careful about those statements because it depends on how you measure genetic identity. Are you measuring it at the level of every single DNA base pair, every single nucleotide in the genome? Or are we measuring it at the level of what genes do they have? Or are we measuring at the level of are the genes they have functionally identical, similar? And those are very different questions. So often when you see those just broad statements, there's no information, no reference backing up what they mean by that. Now, it actually turns out that no matter how we measure it, there's a lot more than we expect. But if we're looking at the level of what genes do they have, there are massive amounts of similarity. So humans and closely related primates, even distantly related organisms, have a much, much higher level of similarity at the level of do they have homologous copies of the same genes. If that's our standard that we're looking at, the answer is, wow, there's a lot more than we would expect. But no matter how we measure it, we end up with this paradox. And the paradox is, based on our early assumptions about how genes work, that there's a huge level of similarity, much more so than we might predict based on this initial hypothesis. And that's the beauty of hypotheses, right? This is the beauty of science also, that initial hypotheses are subject to testing and then revision, right? Or maybe even outright refutation, right? We say, oh, that initial hypothesis was completely wrong, right? So our initial hypothesis of how we build an animal begin to be changed as we looked at this paradox. So humans and chimpanzees basically have the exact same genome, maybe with a very, very tiny level of novel genes, but very, very few. We basically have the same genes. And so we realized that it's not so much about what genes we have, but how those genes are used, when they are used, particularly in early development. A little bit later on, the levels and the genetic networks as adults is important also, but more importantly, like, why do we look and behave so differently than chimpanzees, right? Despite this massive level of genetic similarity, is because of the genes and how they are used during early development. How we build a human is largely the same toolkit, the same tools at the genetic level as how we build a chimpanzee. However, how those tools are used, when they are used, the amounts that they are used in are different. And so it's more about how you use your tools, your genetic tools to build an organism than what tools you have. And that's begun to be worked into our revision of this initial hypothesis. Okay, so we're going to list 8 major principles that we've found, and many of these overlap with those big ideas that we just talked about, these 2 big surprises, right? That proteins can change over time, but there's little effect on function, and that there's this massive amount of genetic similarity for organisms, at least much more than we would have initially expected, and that doesn't correspond. There's this paradox between genetic similarity and morphological similarity. So here are some of the more specific principles that we found that help us to explain this. Okay, so let's define each of these, and we'll probably get into more depth and detail about these in later discussions with specific examples at the genetic level. Okay, so one is this incredible amount of pleiotropy, and we could even call it mosaic pleiotropy, that we find in these toolkit genes. And so this gives us an idea and some implications about the flexibility of genes to evolve and change and take on new function. So this can explain sometimes that that conservation between organisms. So review hopefully, but if not, just learn it. Pleiotropy is where a single gene has more than one job. Or if we want to be a little bit more technical, we could say a single gene has multiple impact on the phenotype of an organism. So for instance, these are some examples in adult organisms. We have genes that, for instance, serve as heat shock proteins in cells. They help to buffer cells against fluctuations in temperature. Some of those same genes are overexpressed in the eye during early development and later development, and form the lens of the eye. And it turns out that many proteins, when fairly pure, can refract light very well. And so oftentimes the genes that are involved in the lens of your eye will have other functions. Okay, so we have a developmental function, and then we have a later kind of a basic housekeeping function. That's an example of pleiotropy. Okay, now mosaic pleiotropy simply refers to the idea that they are expressed in different areas. When you were a kid, I don't know if you ever had to do a mosaic, right? You put together lots of different pieces in different areas to make an overall picture. So mosaic pleiotropy just simply means that in development, some genes are expressed in one part of the body and they have a specific job, and then they may be expressed in a different context, both in forms of where they're expressed in the body and the other genes that are being available, and therefore they will have a different job. And this is true. In fact, many developmental genes have 3, 4, 5, or 6 different specific functions, and therefore we might see them expressed early in development in one area. This is a gene stain where the dark color represents the location of the gene expression at one stage of development, and then later it might be expressed in another stage. And so we might have 2 different jobs for the same gene. Okay, so levels of pleiotropy are a common principle, a theme that we see running throughout developmental genes. Okay, now think about this and what effect this has on the ability of a gene to evolve, right? So, for instance, if you have a gene with a single job, even if it's a very important job, there's going to be some amount of flexibility in how that gene can change and evolve and still accomplish that important job. And we may even have mutations that make it, randomly, just by chance mutation occurs, now it's better at doing its job. And so there can be some flexibility in not only how that gene can evolve and still be conserved, right, which we talked about as that kind of first surprise, but we might also then have some ability for it to evolve and change and take on slightly different functions, and that's advantageous in that environment where the organism is found. And so there's some flexibility both in its ability to evolve and not change, and its ability to evolve and take on new beneficial function. So think of that as a general level of flexibility in the evolution of that gene over long periods of time. Now, if we have a gene that's pleiotropic, if it has 2 specific jobs, or 3 or 4, what is going to be the impact on its ability to evolve? Now, the answer is there's probably going to be more constraint on both of those kind of concepts about how a gene can evolve. There might be less flexibility in how it could change, like swapping out a leucine for an isoleucine might not have an impact on one of the jobs, but it might have an impact on the other job. And so there's going to be much more constraint on pleiotropic genes in a general sense, both in their ability to evolve and maintain function for all of the duties that they have to perform. And also in the idea that if it can change to take on and be better at one of its jobs, we also then need to consider the evolutionary impact that that change has on the other job or jobs of that gene. Okay, so in a general sense, very good take-home message is that the more jobs a gene has, or the more pleiotropic a gene is, the more constraint there is going to be imposed on that gene. And so it's going to be maintained much more over long periods of time, and its function will remain relatively constrained. Okay. Number 2, we can begin to understand ancestral gene complexity by putting together phylogenetics, which is why we did 2 full days on phylogenetics and understanding the principles and mapping and this idea of paralogy and orthology, right? We can understand ancestral gene complexity even if the ancestors, like ancestor X or ancestor LCB here, which is the last common bilaterian, I think, anyway, but whatever name we give that ancestor, we can understand the ancestral gene complexity if we make assumptions and can understand their living organisms. So, for instance, if we look at the last common bilaterian, what were their genes like? Here, this is a Hox complex, but it's just color-coded. And we could say, oh, look, the vast majority of them have this one. This guy has a simpler one, right? And then their outgroup, their closest relative, the bilateria, has an even more simpler. They only have 2 of these many genes. And so by understanding, we'll look at this, and I don't expect you to be able to look at this and tell me exactly right now what the common ancestor's gene was, but understand that we, hopefully this doesn't, spoiler alert, this doesn't ruin your Jurassic Park fandom or experience you have with Jurassic Park, but we cannot look at entire genomes of extinct organisms. DNA has a theoretical limit, and it might be a little bit past this, but survival under ideal conditions, dry and cold, right? So if DNA is dry and cold, it can survive for about 1 million years is what we think. Little tiny chunks of proteins and hints at little tiny bits of genomes might be possible much farther along or more distant than that, but it's very unlikely. So we're never going to have John Hammond reconstructing dinosaurs and us being able to understand their genome. And if we go back to the ancestor bilaterians, right, that's somewhere between 600 million and 1 billion years ago. That's even obviously all the DNA is gone. But we can tell something about the genome of those ancestors that have been extinct for 1 billion years by mapping characteristics that are found in all of their ancestors, looking at outgroups using good phylogenetic principles. We can figure out to a large extent what genes they had, even if we wouldn't know the exact sequence of those genes. Okay, so we're beginning to understand that and understand that ancestral gene complexity evolved fairly early on in the animals and has been passed down as an important trend and correlates fairly well with major innovations in animal form. And we'll look at that in later lectures. Now, this is one that we've hinted at, but I'm going to kind of, hopefully this blows your mind a little bit. Okay. Orthologs, right, review, are copies of genes that can be traced back to a common gene and they diverge at a speciation event. Paralogs are basically duplicated copy of an ancient gene, and so they trace their similarity back to a gene duplication event. So this is an example of orthologs. The paralogs refers to when that redundancy is created by a gene duplication event. So remember, paralogs are created when genes duplicate. And initially, they're going to be redundant. They're going to have the exact same function. Sometimes those are maintained, and sometimes they can then diverge, neofunctionalization, and take on new function. Okay, but for now, for this one, let's just focus on orthologs. So we talked about this gene in mice and fruit flies. In fruit flies, it was named eyeless. In mice, it was named PAC6. There it is. It's an awful name, right? Eyeless is easy to remember, but PAC6 is the ortholog, and it was given a different name because originally they weren't recognized as orthologs, okay? But remember, I told you those are homologous genes, orthologs. One that initiates development, the eyeless gene, initiates development of the fruit fly, and we know that because when we mutate it, when we effectively remove its expression from the genome, we don't get eye development, okay? And the same thing occurs with the PAC6 in the mouse, right? Now, here's what the part that you're like, okay, that doesn't blow my mind, big deal. Here's the part that hopefully will blow your mind a little bit. And this was completely unexpected and illustrates the surprises we talked about early in this discussion, okay? So, mutant versions of fruit flies that have a eyeless gene that doesn't work, if you, and this is one of the beauties of model systems, we can do this in these model systems. If you take the mouse gene PAC6 and express it in place of an eyeless gene that's no longer there, right, no longer expressed, if we were then to artificially express the PAC6 gene, the mouse gene, mouse ortholog, you get recovery of the wild type phenotype. It looks like pretty much a normal fruit fly. So here we have a toolkit gene from a mouse, right? And mice and fruit flies are 600 million years or more diverged from one another, at least 500 million years, right? Think back to the Cambrian explosion. That's when we get origin. It's probably more ancient than that. We just don't have good fossil records from before that. But that's how long ago an ancestor of the a common ancestor of the fruit fly and the mouse existed. Yet despite 600 million years of evolution, we can take a gene from a mouse that builds an eye, put it into a fruit fly that is defective for their copy of that gene, and we recover the wild phenotype. Now that is amazing and incredible and illustrates the conservation of those really, really important genes in development. And again, this is not the case with every single ortholog. Not all of them you can swap in and out. There are changes, often new functions, and so they're not all equivalent, but this is one very good and extreme example of that equivalence. And the cool thing is, right, you're not building a mouse eye, right? So these toolkit genes are basically still part of the toolkit, and it recognizes it as if it was eyeless from a fruit fly. It is that conserved, and it doesn't build a little mouse eye, you know, with a pupil. And we'll look at here just quickly. This is going to be one of the examples we get into in much more detail at the end of the class. It's a cool example, but we'll give you a brief overview of this. So this then goes in and helps us to illustrate the next principle, principle number 4 of deep homology. The eye is a good example of this, but there are dozens of other examples. We'll just use the eye example since we're on it. There are many different types of eyes in the animal kingdom. We're going to look at 3 groups where eyes are very important, but there are other less well-known groups where they have eyes, or at least visual organs, right? So we have the arthropod eye, which we're going to be learning a ton about in the lab. Okay, so I'll save that for others, but in detail, it is a eye made up of individual little lenses and almost like an individual little eye, sometimes thousands of them all put together in a unit to make the entire eye, okay? The other 2 major examples of eyes in the animal kingdom are in the mollusks and in vertebrates. This is a horse eye, but we could put any vertebrate, including humans, in that image. And these eyes are quite similar to one another. They have a crystalline lens, one single lens. They have a retina at the back, and there are lots of other similarities. We won't get into all the details, but they're much more similar to one another than the fruit fly eye is to either of them. But they all serve the same basic function. Now, despite the somewhat similar qualities of these eyes, it was once thought that the mollusk eye evolved independently, because mollusks are very, very distantly related to vertebrates. And there are some other intervening lineages that don't have eyes or have very, very rudimentary eyes. So it was thought that this origin of the eye in mollusks, this origin in the vertebrate, and then certainly amongst the arthropods were completely independent of one another, right? Because they look so, so different physically from one another. Even despite the similarities, there are lots of differences. Just to give you an idea, the focusing mechanism for vertebrates is changing the shape of the lens. In mollusks, the focusing mechanism is changing the shape of the eye, much like an old style camera where you move the lens closer or farther away from the retina, where here we change the shape of the lens. Okay, there are other differences also, but those differences were pronounced enough to think that these were three separate origins. However, upon discovery of this, right, that at the developmental level, the genes that build the eye are the same. That's a remarkable coincidence if it is a coincidence. And in fact, today, we really don't think it is a coincidence. And so this led to this idea of deep homology, that many structures, maybe even, not maybe, almost certainly limbs, like things as different as tentacles and octopuses to arms in vertebrates and segmented limbs in appendages, and certainly eyes are an example of deep homology. That as a visual sensing organ, right, maybe a very simple light sensing organism in ancient ancestor, they are homologous. And so we would expect the very early stages of development to be much the same, like we see given, eyeless and PAC6. That as a light sensing organism, they are homologous. And then the more detailed stages of development, how we get an eye made up of many, many different individual eyelets, so to speak. They're called ommatidia, by the way. We'll get into that in the lab. And the differences between the mollusk eye and the vertebrate eye, that those developed independently. And so that's why we see differences. But at a very deep level, they are the same structure. Okay, so that's this idea of deep homology. And this was not understood and discovered until we found those genetic connections. So a genetic connection tells us that objects or features we once thought were convergent and not homologous at all are actually homologous at a deep genetic level. Okay? Now, the other thing that we found is that despite gene duplication being a very, very important trend among the developmental genes, these toolkit genes, that it's not as common as maybe we might have initially thought. And so maybe the reason for this, why might this be, is because these are really, really critical genes, and changes to them are going to make major differences in the organism. And so any mutation that makes a major phenotypic impact on an organism, if we're just randomly changing it, almost certainly that's going to be a bad thing, right? A random mutation that changes the phenotype dramatically is probably going to be a negative mutation and selected against. Every once in a while, it might be beneficial, but those are probably really rare. So we usually have infrequent toolkit duplication because tinkering with these really, really important developmental genes might have a negative impact. But there are examples of when it does create a benefit, and this is one of them. That when we do have a major change in the toolkit gene, that this case refers to a duplication, and then neofunctionalization of two of the duplicates, that we end up sometimes with a benefit, a major difference, and this is going to be a leap forward or a key innovation. And insects are one example of that. We'll go to this in more detail, so don't worry about the genes or gene names. But we have really good evidence that origin of the hexapod body form, which we're talking about in the lab, is a result of both gene duplication and initial redundancy, followed by a change in both function and location in some of those genes. And we go from a many-limbed organism to a six-legged organism, which is a hexapod. And that's what created this incredibly successful terrestrial group of arthropods, the hexapods. And so to some extent, this, and again, more details to come, but this change, I'm going to go all the way back to our initial example here, right? This essentially explains how we go from a many-legged ancestor, which modern myriapods still show that form, right? And we've got basically the exact same leg on every single segment all the way towards the end of the back of the thorax, if you will, or at least the body trunk. How we go from that to a six-legged organism, we can explain it by duplication of these important developmental genes, followed by changes in the area they are expressed and changes in the function. And so when one of these duplication events and changes in the genes occurs, it often results in a big morphological divergence. We'll look at other examples, but this insect one is kind of a classic one. And think about this, at the genetic level now, we've got great examples of genes changing location, having an impact on where certain structures are formed, which we talked about as a definition of heterotopy. So we are beginning to be able to explain and understand heterotopy at the genetic level. Okay? Next, the modularity of cis-regulatory elements. Now, first, let's define cis-regulatory element. We've kind of talked about it, and I think I even gave you the term, but just to refresh your memory. Near genes, there are these little sequences of DNA that bind to transcription factors. And a combination of that transcription factor and the area of the DNA it binds to helps to regulate that gene. So we call those areas of DNA cis because cis is a term that means near or next to. You might have come across that term in chemistry, where it refers to functional units that are closer to one another in the orientation of the molecule rather than farther apart. But anyway, cis means close to or similar. So cis-regulatory elements are pieces of DNA that are near the gene which they control. And it's very, very unusual to have distantly related regulatory elements. So almost all regulatory elements are cis. Okay, so that's the definition. And so we might have, and they've highlighted them here. Here are the location for cis-regulatory elements right upstream of the gene. The little arrow represents the initiation of transcription. And the start codon is going to be real close to that. And here are the things that do photoreception. However, this is one copy of an eye gene. There are other functions. So iris, not only does it function to help initiate eye development, but it also has other functions. The regulatory elements that help develop eye development are actually in one of the introns, but there also is expression and a function for them in central nervous neurons in the brain. Notice most of these are brain. Some of them are specific to the iris gene, and we don't have in PAC6. But we have all of these different areas that help to regulate expression. And this is a key element because we can have the exact same gene, but then we can have different switches, if you will, right? Different expression initiation areas that can turn it on in different parts of the tissue. So not only does this help us to explain conservation of the gene, but it's also related to this idea of pleiotropy, right? Where one gene has multiple jobs and we express it using different control switches, okay? And so we're beginning to identify these and map these regulatory elements. They're a lot harder to map than the gene itself, so it requires different sets of experiments, a little bit beyond what we're going to talk about in this class, okay? So make sure you know the definition of regulatory elements, and that we can have the same gene being turned on by different sets of regulatory elements that are located in different regions near the gene. And then finally, our last one, we put together these toolkit genes into these vast regulatory networks. So here is and this is probably only partial, but a map of early developmental genes in Drosophila that relate to the orientation of where the head is going to grow, where the tail is going to grow, and everything in between. We have the first initiation of where the head's going to be, and it has many downstream targets. So this is a very highly pleiotropic gene with more than 10, 11, 12, 13, 14, 15, 16, 17 different targets. Okay, and we'll talk about these networks and how we look at them. And then looking at only one of these targets, it then has one downstream target that has, again, many, 3, 4, 5, 6, 7, 8 targets. And again, this is only a partial one, but these vast genetic networks of developmental toolkit genes, one turning on another, turning on 4 more, turning on 8 more, and each one of those we get this very quick and complex set of genes being turned on, and how we do this and at what levels can result in a head growing versus a tail growing, okay? So there are vast regulatory networks of developmental toolkit genes that control differences. And so you could see how even if we had the exact same genes, if we turn them on at different amounts and different levels, or if they are targeting different downstream targets, so we might have the same genes, maybe in a fruit fly, this gene turns on this one, but in a grasshopper, it doesn't. It turns on a different one, or it turns on a smaller subset of genes, and so we end up with a different outcome developmentally, okay? So review those 8 trends. We'll talk about more of them in detail later in the semester, and we'll also give you more specific examples. We'll give you some general examples for some of them. So again, Here is now our working hypothesis, maybe even going towards a theory. So remember the initial theory, which we talked about previously, is that different genes are going to build different animals. So how you build a fruit fly is different than how you build a butterfly because they have different genes. And that was the initial hypothesis, which I'm going to emphasize was wrong. And not only that, but how you build a fruit fly is actually many of the same tools and same genes to how you build a mouse, much more than we would have expected initially. And how you build a fly versus a butterfly might be almost like 99% the same genes, with just different connections and different expression levels and different expression locations. So here is our working hypothesis, and then we might even say this is really more of now going towards a theory. Animal form evolves largely by altering the expression of functionally conserved proteins. This is, by the way, is pretty much word for word from your book. And that these changes largely occur through mutations in the regulatory regions, some in the genes themselves, so it's not 100%, but largely occur in mutations to regulatory regions of these Mosaic Pleiotropy developmental regulatory genes, of target genes that control these vast networks, right? So notice that those 8 concepts are essentially all worked into this one statement, okay? So hopefully you can understand this and then explain little parts of it, at least from a broad perspective right now. Explain how those different parts of it all work together to help us understand evolution of animal form. And then also, this largely explains why the toolkit of a chimpanzee is nearly identical to the toolkit of a human, and how we can build different morphologies with that identical toolkit, and how the toolkit of a chimpanzee, although not identical, is much more similar than we might have expected initially to the toolkit of a fruit fly, and that we can build incredibly different animals from somewhat overlapping toolkits by these same principles. So this general idea explains the differences between closely related species, and also explains the differences, maybe just at a different scale, but the same mechanisms, the same genetic tools, just more or less of the same genes, building very different organisms.